Welcome everyone, and thank you for joining us for today's BrainXell webinar. Today we are speaking about our newly released product, Oligodendrocyte Progenitor Cells. And we are joined by Dr. Clarisse Fligor. She's a research and development scientist here at BrainXell. She specializes in stem cell biology, neuroscience, and iPSC differentiation. She has an expertise spanning complex cell cultures, assay development, and product development. Clarisse has authored numerous peer-reviewed publications and presented her work and research at national and international scientific conferences. She is passionate about developing robust, human-relevant CNS models that advance neuroscience research and accelerate therapeutic discovery. She holds a PhD in stem cell biology and a bachelor's in neuroscience and psychology. Please, everyone, join me in welcoming Dr. Clarisse Fligor.
Thanks, Addie, for that introduction, and I'm ready to get started on today's webinar. I'm going to be talking about our newest product, our oligodendrocyte progenitor cells. So, what exactly are oligodendrocyte progenitor cells? These are also known as OPCs, and these are glial precursors that come from the neural stem cells, and they eventually become these myelin-forming oligodendrocytes. And they are best known for their role of building and maintaining myelin in the central nervous system. However, a population remains in the adult brain and spinal cord, and these OPCs can ultimately be recruited to help to repair any kind of damaged myelin. So they will proliferate, differentiate, migrate to the site, and attempt to repair any breakdown of the myelin sheath. And this happens into adulthood as well, not just in development. So these OPCs, as I said, they persist into adulthood as a renewable progenitor population. So when we're first born, we have our nice cute population of our OPCs that we are eventually going to start to get some myelination, and this continues into through adolescence into young adulthood. However, eventually in middle age, we get our nice full myelination, but in old age, we can start to see that degradation and that breakdown of the myelination. So adult oligodendrogenesis helps to support that myelin repair, maintenance, any type of remodeling. However, in order for this to be successful, these oligodendrocytes need the appropriate axonal feedback from their environment to survive. And so these OPCs that persist in our brain, even so we can see in middle age, we have our oligodendrocytes, we also still have that population of the OPCs. And these need to differentiate into the pre-myelinating oligodendrocytes. And ultimately, only about 80% of them end up dying off because their job is to ultimately myelinate or die. So only 20% of that entire population is fully going to commit to myelination and begin to wrap around those axons. And there's a lot of things that need to go into this for this to be successful. The environment has to be open to it, and you need to get that feedback also from the axons to be able to complete this process. So it is definitely a group effort. And ultimately, the oligodendrocytes have to engage with that axon, receive that activity-dependent stabilization. And this is something that is important, especially as we age. That myelin is going to break down, and the ultimate efficiency of this repair is also going to be less efficient. So as we age, we can see we get this decrease in the population of our OPCs, but at the same time, we're going to get this nice increase in this mature oligodendrocytes, and we have this nice window of myelination. And this is going to be mostly in our adolescence, and it completes when we're about in our late 20s and 30s. And so what's happening in this really tight window here is that our OPCs are going to come and interact with the axon of these neurons, and they're going to begin wrapping around and create these individual segments here, these myelin segments, and they are separated by what is known as these nodes of Remyelination. And what these nodes are is going to be a high concentration of ionic channels, and that is going to allow the action potential signal to jump from node to node, overall improving the speed and efficiency. So it's kind of like the insulation of a wire. So you can see here, this axon is not myelinated, and we kind of get this diffusion of that signal. It's not going to travel very far, and it's not going to travel very fast. However, if it's able to jump from node to node like this, we can get much more efficient transduction of that signal. So this is what the oligodendrocytes are best known for, is making these myelin sheaths. However, there is a lot more to them than just the insulation. They are also going to help to actively regulate the central nervous system function by providing support to these axons, neuronal signaling, and overall metabolic balance in the CNS. And since this myelination continues through adolescence and into adulthood, it's a very dynamic process. But as we age, as I mentioned, that myelin can degrade, either due to damage of the neurons or the oligodendrocytes dying off, and ultimately that's going to disrupt overall cellular function in the central nervous system. And this dysfunction contributes to demyelination and neurodegeneration, which can be further exacerbated by diseases such as Alzheimer's, multiple system atrophy, ALS, or even injuries such as stroke or head trauma. But since a population of these OPCs remain in the adult brain, remyelination is possible, though it is often incomplete. So it is often incomplete and also inefficient. And the presence of OPCs alone does not guarantee that repair. So the remyelination is going to require this coordinated effort of multiple cell types to clear that myelin debris. So here we have some type of injury with myelin debris, and then we need to clear away all of that debris. And once the OPCs are recruited to the site of injury, they must then differentiate into the myelin-producing oligodendrocytes, appropriately engage with the axon, receive that proper feedback, all in order to survive. And so this failure can kind of occur at multiple steps, and it's even more difficult in a chronic disease or inflammatory environment. And just because the OPCs are healthy and ready for the job, if any one of these other cell types, such as the microglia or the neurons, have difficulty, then the repair will not be as successful. And this is why it's important to develop robust human-relevant models to identify where this repair fails and also evaluate strategies that promote successful remyelination. So once we understand what the OPCs need to do in vivo, what they are naturally doing, the next challenge is building those human systems where we can then model those steps in a controlled and disease-relevant way. And a great tool to do this, to create these translational models, is using human-induced pluripotent stem cells. So we can take a healthy patient, take their cells, and reprogram them back to pluripotency and differentiate them into almost any cell type in the body. Alternatively, we can then use gene editing to induce some type of mutation that is relevant to a disease, and this creates an isogenic pair. And this helps us to isolate these mutation-specific effects. And in parallel to that, subsequently, we can also take cells from a diseased patient, use that gene editing to then fix that mutation, and see how these cells act in this pair, and we can kind of really isolate what the exact mechanisms are that are contributing to these different cell types. And using iPSC-derived OPCs helps us to study this human myelination, dysfunction, repair, but also just overall development that we don't fully understand. It gives us a view into how these human cells work in a time of development that we don't usually have access to. So getting into how do we define an oligodendrocyte. So the oligodendrocyte, like I said, it's a very dynamic process, and the different progression of it is marked by characteristic morphology as well as different gene expression. So to start out, you're going to have your oligodendrocyte precursor that will then differentiate into our oligodendrocyte progenitor cells, or OPCs, and these are going to be a highly proliferative population. And they will either maintain this progenitor state or continue forward into this pre-myelinating oligodendrocyte, where they will start to become, you'll see more ramifications come off of their cell body, and eventually they will interact with their environment and wrap around the axons. So along with this change in morphology, we can also mark these different stages by different expression. The one that we are most fond of is this O4 expression, which is going to be very strongly expressed in that progenitor cell. So once they are fully committed to being OPCs, they are going to have this strong O4 expression. And as it further progresses into producing myelin, we are going to see the appearance of these myelin-associated genes such as PLP1, MAG, MERF, MBP, or MOG. So as we can see here in this qPCR, our cells that we produce here at BrainXell initially have a high expression of this OLIG2 and MBP expression, but we can also see that they have the different marker expression of that MERF, CNP, GALC, and MAG, all of these different markers that are going to indicate progression towards differentiation and suggest that this early myelin association maturation, there is potential for that. So this is actually what our OPCs look like. If you were to order a vial from us and thaw them after 24 hours, they're going to have this really cute morphology. They're going to be some bipolar, tripolar cells that are going to have this strong O4 expression. So we can see that with this live O4 staining. It is important to do the O4 staining live because once you fix the cells and stain them for O4, everything is going to light up. So we make sure that you are doing this O4 staining live. So we are able to see the nice morphology, which cells are O4 positive. But we can also further quantify that using flow cytometry. So we have greater than 98% of our cells are going to be O4 positive. And furthermore, we maintain that O4 expression. So we can see at day 0, if we let these proliferate through day 7, we can see that we maintain that similar O4 expression. And these individual dots here are actually different lots of our OPCs, so we can see that we have a really consistent and robust expression profile. And beyond just looking at these graphs, I hope that you can appreciate if we actually look at them on the plate. At day 1, we have some really cute morphology with the obvious O4 expression that is going to be maintained, and you can see the proliferation actually happen over time as we see more cells in this field of view. And it is important that they actually maintain this proliferative capacity because they are a progenitor cell. So they are going to have to expand before differentiating into the mature oligodendrocytes during development, but also during repair. So what we did to visualize this is we did some EDU staining. So that is going to be incorporated into the actively proliferating cells. We treat them with it for 48 hours after thaw, and we either grew them in our proliferation media or we added our maturation supplement. And I hope you can appreciate that the proliferation media, we had a much higher percentage of those EDU positive cells, so a much higher percentage of those cells are going to be actively dividing and proliferating, and that decreases when we add the maturation supplement, which is what we would expect. This is consistent with that transition towards differentiation. So not only are they capable of proliferating, but they are also capable of coming out of that proliferative state and starting to transition towards differentiation. So ultimately, some people either want their O4 positive OPCs, or they want these MBP expressing more mature oligodendrocytes. And what's great about this is we can actually see this transition happen if we do some staining. So we have our live O4 staining, and these O4 positive cells are going to acquire this MBP expression after about 7 days in maturation conditions. And you can see that we get this nice branched morphology in these MBP positive cells compared to the O4 positive cells. The O4 positive cells are still going to be there, but you can start to see that just within a week we are beginning to transition into this more mature state. And this is further reflected here with this qPCR. We can see that these earlier expressed markers, such as that OLIG2, MERF, CNP, are going to either decrease or remain about the same, but the markers more associated with myelin, the MAG, MBP, and MOG, are going to be upregulated in that maturation condition within just a week. So we can see that we are able to get this maturation capacity in this very short time period when these are cultured in monoculture with some type of maturation media. However, growing them in monoculture is not really enough if the ultimate goal is myelination. So OPCs alone are not enough to create myelination. It's the overall surrounding tissue and environment that's going to determine the overall success. The monoculture of OPCs is still good, still useful for characterization and looking at proliferation, but if the ultimate goal is myelination, it's going to require some intricate cell-to-cell interactions with neurons and other glial cells. The oligodendrocytes need to wrap the axons, engage with the axons, and the other glial cells can kind of shape the overall maturation and the repair environment. Something else I want to mention is that it's not only important for these oligodendrocytes to mature, but the overall myelination of a neuron can also help that neuron mature. You're not going to have a fully mature firing neuron without that myelination. So the way that we can look at this is looking at co-culture. We can introduce these other neural and glial cells that interact together to make myelination successful and better support the maturation and myelin formation. And we can actually do that with the cells that we have here at BrainXell. So as I mentioned, oligodendrocytes are going to be an important support glial cell. And what we see is that when we co-culture with our astrocytes, shown here in magenta with the GFAP, and then we have our pre-myelinating or beginning to mature oligodendrocytes with the MBP expression, we see that we get this nice enhanced morphology with a lot of branching. Usually we would just see these kind of flatter myelin sheaths, but we see that when we co-culture them with astrocytes, we get this nice complex morphology that is not usually observed when they are cultured alone. And so this shows us that the glial support may provide the trophic and extracellular cues needed to improve the overall OL maturation. So you can see here in multiple different instances, we have these nice arborized, more mature morphology when cultured with the astrocytes. However, as I mentioned, it's not just the astrocytes that are important. We also want to see how they interact with the neurons. They need that feedback from the neurons ultimately to survive. And what we see when we culture these together is that the OPCs kind of use these axons from the neurons as almost like a jungle gym or as like a roadway for them to explore the environment and wrap around. We see that if we zoom in here, we can see that when we co-culture with our neurons, we're able to support this really nice axonal engagement, not usually observed in that co-culture. So you can see here that it's actually kind of grabbing and following up along this neurite and really interacting with the environment. You can see it kind of wrapping around. So it's this really cool dynamic process that would not be possible if you just looked at one cell type alone in culture. So this neuronal co-culture not only just allows for the cells to interact with each other in the environment, but it also is going to promote the maturation of these oligodendrocytes. So if we are to co-culture our cells with different neural cell types, we either have our cortical glute or spinal motor neurons. We can see that so here is the OPCs alone in the blue, and we can see that we get this nice increase in these myelin-associated markers compared to the monoculture when we co-culture them with the neurons. And these markers that I'm showing here, this PLP1, MAG, MOG, are these different genes or proteins that are going to be important in the actual myelination. So here we have our neuron, and here we have our oligodendrocyte, and we can see that we have our MAG, MOG, PLP, MBP, all of these kind of distributed throughout that myelin sheath. So that's why these are our myelin-associated proteins that we want to see that increase in. So that kind of tells us that there is not only maturation, but hopefully some interaction with those cell types as well. And both our glutamatergic and motor neuron co-cultures are going to kind of shift those OPCs towards this more mature profile. So we can see here, once again close up, that when we have that co-culture, it's going to promote this myelination. We're going to have this strong MBP expression, we're going to have that nice interaction with the axons, And so this is telling us that we really do need all of these cell types together to really mimic what is happening in the body, but also to mature these cell types as well. So why just stick with co-culture? Why just do neurons, or why just do astrocytes? Ultimately, we can combine all of these cell types together and look at it in a triculture. So the co-culture is going to be great if we just want to look at really specific pairwise interactions, but we can expand on that triculture, quadculture, all these different multiple cell types together to create this multicellular context. So the neurons are going to help to provide that axonal support that's necessary for the engagement, alignment, the wrapping of the OPCs. And the OPCs are obviously going to provide their myelinating sheaths. They're going to provide that homeostasis, that overall trophic support, which is also going to be further enhanced by the astrocytes that are going to provide that environmental support. So all of these cell types together, if we look at them in this triculture model, is going to help better capture this multicellular context that is needed for successful myelination. So if we look at these three different cell types, we have our MBP positive OPCs here in green, and then our neurons stained for beta-3 tubulin in red, and our astrocytes with this GFAP positive in magenta. Merge them all together, we get this really cool interaction of all those different cell types, this really engaging environment that allows them to kind of make decisions for themselves and interact with each other, move around, and kind of act more like they would in the human body. And if we look closely, so this is just plated down in a dish, we can see that when we put all three cell types together, we get the advantages of both. We get this more complex morphology that you would usually see when co-culturing with the astrocytes, and we get this axonal engagement. You can see the wrapping, and they kind of like grab on and interact with these axons, and so they're able to kind of act how they're meant to, right? Their purpose is to myelinate or die. And so this ultimately allows them to do their job and also receive feedback from the environment around them. And we can use this as a tool, as a screening assay. And so looking at all three of these cell types together, we can actually use this to look at different pro or demyelinating treatments. So if we were to put these different treatments in the culture, we can do this for a couple of weeks, and then we lyse the cells, and we're able to measure the amount of MBP that we get out. And we can actually see these treatment-dependent changes of MBP, showing us that we're getting this biological response, this modulation, and it's supporting the use for this to be an assay in development, and also for high throughput drug screening. We can see that they are appropriately responding and interacting with their environment. So this is really exciting because it provides a nice scalable platform that we can evaluate these different compounds that can either enhance or also disrupt that maturation and overall myelin output. And what's really great about this is we have the promyelinating, we have the demyelinating, and so once we demyelinate, we can then screen once again to see how we can kind of repair the damage that was done, how we can contribute to that promyelination, and how we can demyelinate and help to kind of mirror what is happening in the body with either disease or injury. And this is becoming more and more relevant as the NIH has recently had this really big push. The new approach methodologies has been this big movement in the scientific community to ultimately replace or decrease the reliance on animal models. So the NIH is really pushing for human relevant tools to either improve disease modeling, drug screening, or other translational applications, and reducing that reliance on animal models. This doesn't mean that they're trying to ultimately replace them. There are still obviously benefits of looking at a full in vivo system, but maybe this can be added to the pipeline. There's a lot of clinical trials that fail once they actually get into human cells, because there is a difference ultimately between other animals and humans. And this kind of gives you the chance to do some type of screening or kind of narrow down some of the choices before moving into in vivo and then ultimately moving into human clinical trials. So these NAMs are important because they're going to better capture that human biology, those cell-to-cell interactions. We can do disease modeling with our different iPSCs, with our different cell lines. We can do toxicity testing, drug screening, and this is all really controlled, scalable, and quantifiable as well. So it really helps to bridge that gap between in vitro and in vivo and potentially help to increase those overall clinical trial success rates. So BrainCell's contribution to this is we have actually recently launched our BrainBlox. And basically this is our contribution to this, in that we can take all these different cell types that we have in our catalog and we can provide these different building blocks, kind of like Legos. You can take all of these individual cell types and combine them in any type of combination that you want. And we can also create reporter lines or disease lines. And this really helps to model those intricate cell-to-cell interactions in our human cells. And so we can have defined cell inputs. We can decide which cells go into it, what ratios, if you want more of a certain cell type or less of a certain cell type. And we can also scale this either in 2D or up to 3D assays. And this is going to be important, especially for the OPCs, to kind of study that myelin development, dysfunction, and repair. So I mentioned that we can scale up from 2D to 3D. So why would we want to move to 3D? And if you think about the development of the human body, it does happen in 3D. 2D is obviously much easier to do, and that is just all on the dish. However, 3D has some really great advantages in that it does a lot of the work for you, right? It recapitulates that kind of interaction of the cells. They're going to make their own extracellular matrix. They are going to arrange themselves how they want, and you can culture them for a much longer time. But beyond just those advantages, what we see is that we actually see increased maturation as well. So if we look once again at our myelin-associated markers, we look at just our OPCs. So this MAG, MBP, and MOG are probably the most well-known. We can see that in the 2D monoculture, we do still see an increase when we have the maturation media, but that is further increased when we do this 3D monoculture. So when we aggregate them together in spheres and culture them as floating spheres, which is already exciting. It's great. We can culture them for much longer. However, once we then add in the triculture, so adding in the multiple cell types now, we can see this really significant increase in these myelin-associated markers, which is telling us that this multicellular environment is enhancing that maturation and the myelin output. So we can see that we have this significant increase in these different myelin-associated genes compared to the 2D monoculture, but also the 3D monoculture and the 3D triculture is ultimately going to give us that highest expression. And this is interesting because the 3D triculture does have the highest expression of this MAG, MBP, and MOG, but the overall cell input is actually less compared to these other samples because that sample is also going to include neurons and astrocytes, showing us that we are just having even more of that expression if we were to normalize it back to the overall cell input. So this multicellular environment really enhances that overall oligodendrocyte maturation, and we're able to culture them for much longer. As I mentioned, myelin is a very long process. Myelination happens, in gestation through your adulthood. And so you actually do need time to really mimic that. And 3D seems to help to accelerate that and recapitulate what is happening in the human body. So I just want to point out that this MAG and MOG, if we were to look at this myelinated axon in vivo, we would have this MAG expression on the inside closest to the axon, and then MOG is going to be this later expression on the outside once that myelin is actually fully wrapped and compact, and that's going to show us that terminally differentiated true mature oligodendrocytes. So that's why this is really exciting to see all of these not only upregulated, expressed and upregulated, but significantly when they are all together with multiple cell types. So Speaking of our BrainBlox and moving into 3D, we are excited to be working on a 3D platform for myelination. We're able to create high throughput, really consistently sized aggregates. We can either do our 3D monoculture, so OPCs alone, and we can control the size of them based on the number of cells that we put in. So as you can see, we get this nice kind of linear increase in spheroid size, depending on how many cells we input into it. So you can really control and customize what you want. And we can further enhance this by adding in our other cell types, so our astrocytes and our neurons, and combining these all together into that spheroid to create this kind of multicellular CNS organization. And I think the most exciting part of this is that we're able to culture them for much longer, as I mentioned. So one thing I want to point out is just this overall kind of anatomy. So if we were to look at this myelinated sheath of the axon and the oligodendrocyte wrapping around it, if we were just to section through that and look at it head on, what you would see is the axon and then these really tightly wrapped concentric layers of myelin. And if we culture our 3D oligodendrocyte triculture for up to 8 weeks, we actually did see the same type of wrapping. So you can see here in this section, we have multiple areas where you can see these concentric rings of the myelinating wrapping around structures within this spheroid. And the reason that we know it is myelin is that it's so tightly packed that the light can't get through. And so that is what tells us that this is a nice myelinated area. And so seeing that ultrastructural organization really supports that these oligodendrocytes are truly maturing in this 3D environment. And this is something that is not usually seen in a 2D environment. So if we section through one of these 3D spheroids with all these different cell types in it, and we use this transelectron microscopy to look at these really high magnification, we can see that this truly is a dynamic process. The membrane organization is going to range from these looser wrapping. You can see multiple layers that are really apparent here, but then we also get this really nice compact structure of these multiple different layers. And this is ultimately what you want to see for that true mature myelination, is these really distinct tight rings. And so this was really exciting to see that we were seeing this within our structures after just 8 weeks in culture. Usually this takes months in vitro. It takes years in vivo, but we're seeing it within just 8 weeks. We can start to see this process begin to happen. So this is important as a translational application. Why do these matter ultimately for drug discovery? So that marker expression alone isn't able to confirm meaningful or functional myelination. Just having that MBP expression or MOG expression doesn't tell us if these cells are truly myelinating. And these 3D systems, such as these myelinoids, can really help us assess that overall oligodendrocyte maturation, that axonal engagement, and ultimately that myelin ultrastructure. So these really controlled human models help to preserve those cell-to-cell interactions, and they also are going to reduce any of those extraneous variables that can complicate interpreting your results. And ultimately, you are left with quantifiable readouts that are going to support compound screening, disease modeling, and help you prioritize your therapeutic approach. And there are multiple different ways that this can be used, whether you're just looking at neurodegeneration, the overall intercellular dynamics, even adaptive myelination. As our body moves, as we learn, as we grow, myelination is going to respond to that. And that is important in development, but also in disease and in aging. And furthermore, we can look at different environmental exposure, different therapeutic compound screening, and we can genetically manipulate different cell lines to look at the interaction of different genes or proteins, and ultimately kind of create this more human relevant approach to studying disease and development. So there is no single true model that's going to answer every single question. The goal is to ultimately choose the simplest system that's going to capture the relevant biology needed for the assay. And what I mean by that is that just because 2D doesn't show ultimately true myelination doesn't mean that it's not useful. it's still going to be useful for initial screening, that more high throughput, and also looking at reproducibility. And then you can further move on to 2D co-cultures and tricultures to study those really important cell-to-cell interactions. And ultimately, you can combine this all into this 3D myelinoid to further validate that advanced maturation, any type of therapeutic potential, and ultimately that myelin ultrastructure. So, in summary, I hope that I have shown you today that our human iPSC-derived OPCs demonstrate a robust oligodendrocyte lineage. They have strong proliferation and maturation capacity. And in monoculture, they maintain that O4 expression and then acquire that MBP expression after maturation. If we are going to further combine that with other cell types, we can integrate neurons, astrocytes, and better capture that multicellular interaction involved in myelination. And ultimately, if we combine that all into 3D, we can get that long-term maturation with evidence of wrapping and compact myelin structure in a very short amount of time. So in conclusion, overall, oligodendrocytes are really essential for the central nervous system function through not only myelin formation, but metabolic support and also long-term axonal health. And these OPCs are going to provide a repair-capable population, but the remyelination ultimately depends on the successful maturation, axonal engagement, and a supportive multicellular environment. So these human iPSC-derived OPC models are going to allow these factors to be studied in a more controlled and disease-relevant system. And increasing that model complexity from monoculture to co-culture, triculture, ultimately moving into 3D, is going to enable those different biological questions to be addressed. So together, these platforms support scalable screening, deeper validation of therapies, and overall help us aim at promoting myelination and repair. Thank you very much for your time, and I'll hand it back to Addie.
Clarisse, thank you for that great presentation. Not only speaking to our new product, Oligodendrocyte Progenitor Cells, but also to our new offering, BrainBlox, which we are very excited about, and our NAMS initiative, as we continue to expand our portfolio and our NAMS portfolio. If you have any questions, please see the link in this video and the text below. There's a way to directly contact Clarisse and our team at BrainXell. We are happy to answer any questions. There's also a link so you can see more information directly on our website, and we look forward to continuing to serve your research and drug discovery needs. And thank you for joining us.